Dear students, in the following lecture I would like to talk to you about the liver, bile ducts and the pancreas. First of all, let's see the liver. The liver, or hepar in Latin, the biggest gland in the human body. The average weight of the liver is 1.5 kilogram and is located under the diaphragm. The bigger right lobe of the liver can be found in the right side of the abdominal cavity, just below the right ribs, and the lower and the little left lobe would be on the left side of the abdominal cavity. In normal physiological case, the lower border does not exceed the ribs. In case of alcoholism or hepatitis, the liver will be bigger. This is the liver enlargement, and thus exceeds the ribs on the right side. The liver has two different surfaces. The diaphragmatic surface is covered by the diaphragm, and one of the double layer of the peritoneum divides into two lobes, the bigger right lobe and the smaller left lobe. The inferior surface face to the abdominal organs or viscera, so the name of the inferior surface, visceral surface. In the middle of the visceral surface we have a gate of the liver or porta hepatis. The main structures of the porta hepatis are the following. In the middle of the porta hepatis we have a portal vein with purple color on this picture. The portal vein comes from the GI tract, comes from the stomach, the spleen and the small and large intestines. The smaller red structure will be the proper hepatic artery, which comes from the common hepatic artery of the abdominal aorta and carries the oxygenated blood into the liver. And you can see the hepatic duct with the green color carries the bile produced by the liver, and the hepatic duct carries the bile into the gallbladder. The gallbladder is visible on the visceral surface on the right side. And these are the different impressions of the neighboring organs. For example, the stomach impression on the left lobe, renal impression and colic and suprarenal impressions on the right lobe of the visceral surface of the liver. And finally, in between the right and the left lobe on the visceral surface, we have a last portion of the inferior vena cava. The inferior vena cava pierces the diaphragm, and after piercing the diaphragm, opens into the right atrium of the heart. Let's see the basic knowledge of the histology of the liver. The histological unit of the liver is the hepatic lobule, which is a hexagonal shaped structure. In between the hexagonal hepatic lobules we have a few connective tissue in human. In the center or middle part of the hepatic lobule you can see the central vein, and around the central vein we have a hepatocytes. The hepatocytes form rows, and all the row of the hepatocytes will reach the central vein. In between the hepatocytes or hepatocytes rows, we have a sinusoid capillaries. The sinusoid capillaries contains blood, of course, and this blood in the sinusoid capillaries flows into the central vein of the hepatic lobule. Because the hepatic lobule is a hexagonal shaped unit, it has 1, 2, 3, 4, 5, 6 corners. In between the lobules, approximately at every second corner of the hepatic lobule, we have a portal triad. So after this name, the portal triad will contain 3 different structures. The biggest structure in the portal triad called interlobular vein. Interlobular vein belongs to the hepatic portal venous system. The smaller structure in the portal triad called interlobular artery belongs to the proper hepatic arterial system. And finally, the smallest structure, the bile duct. The bile duct formed by the beautiful cuboidal epithelial cells, and the bile duct will carry the bile into the hepatic duct. So interlobular vein, interlobular artery, and bile duct. You can see the histology of the liver in bigger magnification. This is the central vein in the middle point of the hepatic lobule. These are the hepatocytes with round nuclei, and between the hepatocytes this gap-like structure will be the sinusoids with blood. And now let's see the hepatocytes and the main characteristics of the hepatocytes. So the hepatocytes is a cuboidal shaped cell, epithelial cell, so it has 6 surfaces. But the surfaces has 2 types. One of the surface face to the sinusoid capillaries. You can see this surface here. This is the blood pool of the hepatocytes. On this surface there are lots of microvilli, and the microvilli will increase the surface for the absorption. The microvillated surface of the hepatocytes face to the sinusoid capillaries, or we have an extremely small space between the fenestrated endothelial layer of the sinusoid capillaries and the microvillated surface of the liver. This is the disce, space of disce. So, the microvillated surface of the hepatocytes faces to the space of disce. Space of disce contains blood. The other surface of the hepatocytes faces to the neighboring hepatocytes, and in between the hepatocytes we have a bicapillaries. The bicapillaries do not have a wall. The walls of the bicapillaries formed by the cell membrane of the neighboring hepatocytes and contains bile, of course. So this is the bile pole of the hepatocytes. This picture you can see the bicapillaries only with silver nitrate impregnation. So the bicapillaries build a network within the lobules. The blood in the sinusoid flows from the peripheral part of the hepatic lobule into the central vein. In contrast, the bile in the bicapillaries flows from the middle part of the hepatic lobule into the peripheral part, into the bile duct. And now let's see the circulatory systems of the liver. There are 2 circulatory systems can be found in the liver. The oxygenated blood comes from the proper hepatic artery, and it has many or lots of branches in between and around the hepatic lobules. The portal vein comes from the, as I mentioned, from the stomach, the small and large intestines, and from the spleen. And the blood of the portal venous system extremely rich in nutrients. The branches of the portal venous system can be found between and around of the lobules, and finally the 2 systems meet with each other in the sinusoid. Therefore, the blood of the sinusoid capillaries is a mixed blood. The blood of the sinusoids flow into the central vein with high level of carbon dioxide, and the central vein continues as hepatic veins, and finally all the hepatic veins drain into the inferior vena cava. Absorbed nutrients reach first the liver, and only after the systemic circulation orally given drugs too. You can see one of the functions of the liver. So the liver products the bile, the liver products the blood protein, for example albumin, globulin and clotting factors. The liver has important roles in the lipid and carbohydrate metabolism, for example can storage the glycogen. Detoxification function is important in the medicaments, and the liver can storage one of the vitamins, for example vitamin A and B12. And now let's see the portocaval anastomosis and the clinical importances of the portocaval anastomosis. What does it mean? Anastomosis means connections. So there are small connections between the two major venous systems in the human body. One of the venous system, the cava system, cava superior and inferior, and the other venous system in the body, the portal venous system. So we have lots of small connections or anastomosis around the esophagus, between the superior vena cava and the portal venous system, around the rectum and the abdominal wall and in the retroperitoneum. Clinically, the first and the second anastomosis will be the most important. When portal blood flow is hindered, these connections will be enlargement, and the consequence of this enlargement, the bleedings. For example, around the esophagus, we have an esophagus varix or varicosity. So this is the enlarged or enlargement anastomosis, and it has a bleeding. Or in the lower part of the rectum, the enlarged vein is called hemorrhoids. It's visible in the final portion of the rectum. So once again, the clinical importance of the portocaval anastomosis, the bleeding from the portocaval anastomosis, and the blood pressure in the portocaval anastomosis and the portal venous system will be increased. And the other consequence of the increased portal pressure, for example the ascites, so in this case extremely lots of fluid accumulate in the peritoneal cavity. Let's go to the gallbladder. So the gallbladder is located on the visceral surface of the liver on the right side. The name of the duct of the gallbladder, the cystic duct or duct of gallbladder. Inside the gallbladder, the inner surface of the gallbladder formed by the absorbing epithelium, so microbillated columnar epithelial tissue and concentrating the bile. So the bile produced by the hepatocytes, and this bile has lots of fluid, water. In contrast, the gallbladder concentrating the bile, so the bile in the gallbladder has a few water. The bile ducts divides into two different groups. We have intrahepatic bile ducts, this will be the bile capillaries and the bile duct, and the extrahepatic duct contains the cystic duct and the hepatic duct, and after meet with each other, this is the common bile duct. So the common bile duct formed by the cystic duct from the gallbladder and hepatic duct which comes from the liver. The common bile duct pierces into the head of pancreas and opens into the duodenum together with the main duct of the pancreas. So this point shows the common opening of the common bile duct and the main pancreatic duct in the middle part of the duodenum. Pancreas, the following topic. The pancreas anatomically divides into head on the right side, surrounded by the duodenum, the body, this is the middle part of the duodenum, and finally the tail. The tail reaches the spleen. The body and the tail of the pancreas are located in the retroperitoneum, so the posterior part of the abdominal cavity behind the parietal peritoneum. Around the pancreas there are lots of extremely important organs and structures. For example, behind the body you can see the inferior vena cava and abdominal aorta and the superior mesenteric vein and artery. Histology of the pancreas. The pancreas contains exocrine and endocrine glands, 2 in 1. The exocrine glands of the pancreas are the serous glands, and the serous glands of the pancreas will produce the digestive enzymes, for example trypsin, lipase and amylase. The trypsin digests the proteins, the lipids digest the lipid acids, the amylase digests the carbohydrates. Opens together with the common bile duct, so the main pancreatic duct opens together with the common bile duct into the duodenum, and the enzymes are activated here only. So all the digestive enzymes of the pancreas produced by the serous glands in inactive form. When activated already in the pancreas, this will be the acute pancreatitis, and the pancreas digests itself. In case of chronic pancreatitis, what does it mean? There are not enough enzymes with digest problems, and different medicaments contains enzymes that help digestion. The other name of the serous glands is called acinus or acini. The endocrine part of the pancreas represented by the Langerhans Islands. The Langerhans Islands can be found in between the serous glands of the pancreas. So the Langerhans Islands is the endocrine part, so do not have duct system and produce hormones, and these hormones going to the blood circulatory system. The A or alpha cells of the Langerhans Island produce the glucagon. The B or beta cells produce the insulin, and the type D or delta cells will produce the somatostatin. The insulin decreases the level of the blood sugar. One of the most important clinical importance of the endocrine part of the pancreas, the diabetes. In type 1 diabetes, the beta cells or B cells degenerate and there are no insulin. The insulin treatment helps for the patient. In type 2 diabetes, in older ages and more common than the type 1 diabetes, the tissues are not so sensitive for the insulin. For example, there are no insulin receptors on the surface of the cells in the human body. Lots of drugs to induce the insulin production. Okay, and one picture about the portocaval anastomosis. So this is the normal liver, and for example, in case of cirrhosis, reasons of the cirrhosis, the alcohol and the hepatitis, the structure, the inner histological structure of the liver is changed, and the pressure in the portal vein increased, and the anastomosis enlarged, and the consequence of this fact, the bleedings in the portocaval anastomosis. Thank you for your attention.